Very good morning, dear students. Let us talk about the transgenic plants in paper 4 of zoology. Where transgenic plants means they are the plants where the DNA is modified, genetic material DNA is modified by a technique called genetic engineering technique. So let us know what is genetic engineering technique. See, transgenic plant, genetic engineering is a technique where the gene content or the DNA of one plant is transferred through vectors to the other plant in order to get desired characteristics. That is genetic engineering. So with this technique, transgenic plants have been produced. A transgenic plant is a plant where it is referred as a modified plant, where in these plants the genes are transferred from one plant to the another plant through a genetic engineering technique, as I told you just now. The purpose of producing this transgenic plant is to obtain the desired characters which are not present in the earlier plant. What are those desired characters? That is in terms of yield and also in terms of quality. See, with regard to the production of transgenic plants, we have two methods. The number one is the agrobacterium mediated gene transfer method, and the second method is gene gun method. In the first one, that is in case of agrobacterium mediated gene transfer method, agrobacterium, as you all know, it is a bacteria, where this bacterium acts as a pathogen which causes the disease, where it is known to cause a disease called as a crown gall disease in members of the family Rosaceae, where after the infection is over, the symptoms are seen in the form of swellings just at the above the soil level, that is at the lower node of the stem regions. After infecting the plants, the pathogen Agrobacterium tumefaciens, which is responsible for causing this disease, transfer the genetic material which is carried by it, which when it acts as a vector, and it will be transferred to the genetic material and it will be incorporated to the genome of the host plant. As a result, the host plant already has some genetic material, and that genetic material is being mixed with the newly carried genetic material. So the two genetic material mixed together causes a disease, and it shows different characteristics. With regard to the gene gun method, it is a method where the desired gene is coated in a gold or a tungsten particle and bombarded into the plant cells, or incorporated into the plant cells through the vectors. When once it is incorporated, the sequence into the plant cells, which can be proliferated by a tissue culture method. So with regard to the applications of transgenic plants, these plants are basically produced in order to get the following benefits. It shows a resistance to the biotic stress and also abiotic stress, and transgenic plants will have a very high yield, and they also have a very high quality, and there is a nutrition level is also increased in transgenic plants. As a result, as they have very high yield and they have very high nutrition, the commercial value of the transgenic plant is also improved. As a result, the farmer gets benefited and the economy is increased, and the transgenic plants also act as bioreactors. So these are the following are some of the examples for transgenic plants, where golden rice, BT cotton, flower sour, and in addition to these plants, potato, species of potato, species of corn, papaya, and some species of maize and pumpkin also have been modified as transgenic plants in order to obtain a better yield, increased yield, and also resistance to diseases, and also had to have high commercial value in order to help the grower. This is all about the transgenic plants. With regard to the cryproteins, these cryproteins is nothing but a crystal protein, and they are always poisonous to insects, and they are expressed by cry gene. Bacillus thuringiensis is another bacterium where it synthesizes this cryprotein when this bacteria is in inactive state at pH when it alkaline, during alkaline pH. The insects helps the activation of the cryproteins. Cryproteins are non-toxic to human beings and also some animals, but they act as a toxic substances as far as insects are concerned. Let us see how these cryproteins are produced, that is production of cryproteins. The bacterium Bacillus thuringiensis synthesizes this cryprotein. Except for few insects, as I told just now, this bacterium is considered to be a non-toxic to for few plants and animals, but it is toxic to insects. So it is host specific in its activity, and cryproteins are exploited by man by producing insect resistant crops in Bt cotton and Bt brinjal. So it is all about the cryproteins. Thank you.